Well, hello, this is Miss Dagenford, and we are going to be learning today about amino acids. And amino acids are classified by two unique functional groups. Over on the left here, we have an amine, and you can see that it is a nitrogen and two hydrogens. And then over on the right, we have the carboxylic acid. This is an organic acid. It's a weak acid, and it's characterized by that double bonded oxygen and then the oxygen hydrogen hydroxyl group. So some familiar components. We've seen the acid part before. The amine is new. Now you might be thinking, what in the world is this R? I've never heard of this R. Well, the R is the side chain. And uh, some books, some students like to think of it as the rest or the remainder, but really what this is it's similar to the X in algebra. It's a variable. It can be multiple things. The R group is what specifies the type of amino acid, the identity, the specific amino acid. So the R group is unique to the amino acid that we're talking about, and there are going to be 20 that we will see. Now, they found close to 500, but really it's the 20 that are encoded through the genetic code that really are what we're going to be looking at. So there are 20 of these different side chains. And so notice glycine here has a hydrogen, aspartic acid has this carbon, and the COOH. Now that is the carboxylic acid, carboxyl group. And so you can see a lot of these different functional groups around. If we just scroll to the right here, I have a little chart that shows, and your book has one like this. Now, just a quick note to relieve any panic, you do not need to know the specifics of these. One thing that you need to understand is the general structure of amino acid and then how these side chains influence the structure and then the importance of that structure. And this is not going to be a one-time thing. We're going to be looking at proteins all throughout the year. So you'll become quite comfortable with the implications. But again, you do not need to know any of these specific structures. But notice we can see that some of these are non-polar. And if you give a quick glance, all of these, notice these are hydrocarbons. They have no or very minimal they're very neutral. They have very minimal dipole moments. Then we have the polar, which can be, here we have our OH groups, which should become very familiar. SH, sulfur, is very similar to oxygen in the periodic table. Here's another OH, NH2, which is also polar. So there's our polar ones, and then we can even see ones that have full charges. They're ionic. Now, that also means they're acidic or basic, depending on the charge that they carry. So this ends up leading to some of the chemical behaviors of these. Now, so does that mean they're polar or nonpolar? Well, as you just saw, it really depends on the R group, that side chain. Some of them are, some of them aren't. Now, the polarity of the OH and NH groups means that they will be able to form hydrogen bonds. We'll see that in just a little bit. But it's those side chains that also influence the behavior. And what new atom did we add? We added nitrogen. Oops, that's no. And then what we can do with these amino acids, just as we've seen with the monosaccharides and the fatty acids and glycerol, we can actually connect them together. So let's just go ahead and draw a glycine. And we'll draw another one right next to it. And you want to make note of the fact that we're putting the nitrogen and carbons in the same order. We have nitrogen, carbon, carbon. And I like to kind of shorten it to NCC, NCC, and that's very, very specific. We have, and there's a reason for that, and that is because this allows them to form a dipeptide. And at this point, you should be thinking, I bet I see where this might be going. If we take that oxygen and hydrogen, and then this hydrogen, you might naturally start thinking, oh, seen this before. This is going to come together to form water. And then if I were to draw this over here, what this now means is that this carbon now has the ability to join directly with that nitrogen. So let me go ahead and just finish drawing this, and then I'll point that out. Now, it doesn't matter what amino acid we're talking about. This reaction, this dehydration synthesis, can still occur. So let's not forget those hydrogens. So we're talking about this bond right here that forms between that carbon and nitrogen. And this has a very, very unique name. This is now a peptide bond. And we see that for every amino acid can form a peptide bond. Now, what we're going to see is that these amino acids can join together to form dipeptides. So right now I've made a dipeptide. They can form tripeptides. And you can see where I'm going with this. Hopefully we see tripeptides. If we join multiple amino acids, we would get to a polypeptide. And the polypeptides, some of them can function alone as their own protein. So sometimes polypeptides and proteins can be kind of the same. But most of the time, if you're seeing polypeptides, they might interact with other polypeptides to form a larger protein. And what do these proteins do? Well, they can do pretty much anything you can think of. They can be structural functions. They can be enzymes. They can be immunoglobulins. We learned them as antibodies. They can be receptors. They can help form identifying proteins. So establishing what the cell is. They can be adhesion molecules between two cells. So the list is pretty amazing. They can be hormones. So they can cause cells to signal. So what we want to look at, and this is why we're going to see these all throughout the year, is that this comes up again and again and again. So you should be able to fill this in because I actually identified this about a minute ago. The bond between amino acid is called a peptide bond. Now, as a protein is being built, it forms different levels of structure. The easiest of this and the most fundamental is the primary structure, and the primary structure is the sequence of the amino acids. And the sequence of amino acids is as simple as if we have, let's just take 3 different kinds: glycine, proline, and leucine. Well, that's maybe that's a sequence for protein A. Well, protein B could be something like leucine, glycine, and proline. And just doing that is going to now establish that this is a different protein. So the primary structure, you want to think about it as maybe the how we build them together. It's the sequence, it's the order and the type of amino acids. We're going to make a really important connection between this and the genetics in a couple of months. Now, as they come together, the hydrogen bonds that can form between the backbone of the amino acid can form these twists and sheets, and we call that the secondary structure. We have 2 of them. We have an alpha helix and the beta pleated sheets. So here's our alpha, and then our, whoops, I'm sorry, I did this before too. We have our alpha, and these are formed by hydrogen bonds. So we have our alpha helix and beta, and those are Greek letters, alpha, helix, and beta. And I'll show you a picture of this in just a second. Those alpha helices and beta sheets, think about the strand of amino acids starts coiling around. And then as the coils themselves start bending, we get into the three-dimensional shape of the protein. And then if we have multiple polypeptides, 2 or more polypeptides that interact. Now, as they interact, that's going to create this unique shape. This shape is going to be hugely important to its job, and we're going to be seeing that again all throughout the year. So the three-dimensional shape, we find that really coming together in the tertiary structure. So let's look at how this happens. So here we have this little twist, this helix. Here's our helix. And this helix, this is the alpha helix. Now notice, here's our little dots. Let me zoom in on that a little bit. And so here are the hydrogen bonds. And these hydrogen bonds, notice they're the interactions between the backbone of the amino acids. In other words, not the R groups. So these hydrogen bonds are interacting between the oxygen, between one amino acid, and then the hydrogen connected to the nitrogen. So this would have a partial positive, and then this oxygen would be a partial negative. And that's what allows them to curl up and form these unique helical structures. Now the beta-pleated sheets, they are also formed with hydrogen bonds, but they form these flatter ribbons and not the curls. But the importance is it's the hydrogen bonds coming from the backbone, and it establishes these really unique helical structures. So here's another picture. Here's a helix. So this is the alpha helix over here. And then this would be the beta sheets. And again, notice they're kind of causing these folded flat ribbons, and that causes a very unique structure as well. Now, here's a larger picture. This is going to represent the tertiary. And the tertiary structure, notice here would be this would be an alpha helix. This would be an alpha helix. But notice this large superstructure is held together by multiple interactions. Here we see, they call it a salt bridge, but the other way to be familiar with it, these would be ionic interactions, where we have a full charge. This would be a full positive, this would be a full negative, and they're holding that part of the protein together. Here we would have hydrogen bonds. This is a partial negative, here's a partial positive, so that can occur. And they are only showing little bits of it, but keep in mind this could be happening here and holding together these two groups as well, and they're showing there. Now the yellow here, those are called disulfide bonds. Those are sulfur-sulfur bonds. They're very strong. They're covalent. So that really strengthens the protein shape as well. And then we have these hydrophobic interactions. So overall, this group over here might be excluded from the rest of the protein because it is neutral. And that's going to also play a huge role in how these interact within the cell. So all along the protein, we have these little regions of charges or neutrality, which then dictates the shape it's going to be in, and then that will dictate its function. And so that concludes this little introduction to amino acids, and we will talk more about this tomorrow. Have a good day.